Liplo. Once again, let us have this tutorial for biology. In this paper, we are going to answer only questions on the topics that we have covered with Liplo line. So we are to skip other topics that we have not asked. So we are to focus on those questions that we have we have covered till we finish. From there is when we are to answer all the paper. So here, just want to give you a sample on how you should be thinking and you should be answering, and on how you as you are studying on 4 areas to focus. All right, let us start. Define safety. Safety. These are application measures or measures taken to avoid accident in the lab. Measure any tools using glassware in the lab. Mention 2 rules of using glasswares in the lab. So when you say glasswares, there are those apparatus that are made of glass. So how we should use them. The first one is that you must follow a proper instruction guided by your teacher on how you should use those glasswares, and also you should not misuse glassware. For example, ngati glassware yonyamulira liquid yokuisanchira kumanyamulira zinthu zina. So those are some of the rules on how you should be using those glasswares. In an experiment, to find out the effect of temperature on enzyme activity, identify independent variable due to dependent variable. So the experiment is effect of temperature on enzyme activity. Effect of temperature on enzyme activity. So independent variable are variable that do not change, while dependent variable are variable that changes. Dependent ndio madalira, independent sa madalira. So whenever you are carrying this experiment, you are asked to identify temperature that independent, that do not change. So temperature that do not change, this include independent variable. These can include type of enzyme. If you are conducting this experiment, make sure the type of enzyme must not change, but you can change maybe temperature, you can change light intensity, you can change maybe types of you can also change amount of enzyme. So zimene unga synthesis, those are dependent variable, because dependent variable they change, while independent they're the ones that do not change. So types of enzyme cannot change. 2, also you can say amount of enzyme. This topic is under ecosystem, so we are not going to bother that one till we cover that topic. Let's go to this one. Question 3, I think this question 3 we've done. This is under photosynthesis. So the question says the equation below is one of the processes that occur during photosynthesis. So we are being shown water to produce H plus R. So water to produce H plus R. So name the process. Name the process. Let me take you again the question. The question says the equation below is one of the processes that occur during photosynthesis. So one of the process, and they are showing us that water has been split into hydrogen plus R. So name the process. So philosophy is not in my stage away. That's light stage and dark stage. So that light stage material water split to hydrogen and oxygen. So name the process. That is photolysis. The process is known as photolysis, the splitting of water into hydrogen and oxygen. Which stage of the surface is this one? So this stage is light stage. So on the equation, R, it seems it actually is oxygen, since water is being split. Mention any two things that happens to the product R after being produced. Oxygen and catalysis. What it does, it produce. So one is used to form carbohydrates, to make proteins, so on and so forth. Fats, so on and so forth. That's it. So you can see how this question has been asked. So be clever. So the stage here for photosynthesis is one of the stages of photosynthesis, which is a light stage. Part B, let's skip this one, is experiment part, of which we did not cover, but we have to cover it as I promised. The paper 2, my practicals. Apart from carbon dioxide, explain any two other necessary conditions for photosynthesis. Apart from carbon dioxide, so you have water, sunlight. Those are some of the necessary conditions for photosynthesis: water, sunlight, chlorophyll, so on and so forth. Figure shows a cell undergoing one of the stages of mitosis. Use it to ask the question that follows. So look at this stage. Name the stage of mitosis. Actually, we covered this yesterday. So we see that a chromosome are being splitted. They are being splitted and they are at pause, but it has not split. So name the stage, give a reason for your answer. Yeah, so we see that this is anaphase stage, since the chromosomes have been splitted and they've migrated to opposite poles. So name the stage is anaphase. Give the reason that chromosomes have migrated, they have spread and have migrated to opposite poles. Suggest one reason which show that a cell in figure 1 is an animal cell. So I think you should answer this one. Why does this is animal cell? Can you suggest on your own? I will not reveal the answer for that one because yesterday I tackled so much at that part. Okay, let's come to this one. The figure below is diagram of a process that occur in human alimentary canal. So we have large fats. Look at the diagram. Large fats, globules. They have been split into small parts of globules. So large part, small droplets of fats. So coming back to the alimentary canal digestive system, the process is let us answer the question. Name the process above. So the process is known as emulsification. Remember, bio is the one that breaks the large droplets of fats into small droplets. Identify the substance marked Y. Y. That's bio. So the substance labeled Y is bio. What type of digestion is this one? This one is physical digestion. Don't bother to say chemical digestion. It is physical digestion because physical digestion. What happens is that large droplets are broken into small droplets. In chemical digestion, there is an action of enzyme. Enzyme. Explain what could be the effect of replacing hydrochloric acid with sodium hydroxide in the stomach. Give 2 points. What is the function of hydrochloric acid in the stomach? Hydrochloric acid in the stomach will break down bones, neutralize bones, create medium for work of enzymes. Those are some of the commercial kill bacteria and germs. Ngati tajosam hydrochloric acid, pigamos alkaline, sodium hydrochloride, that's alkaline. What will happen? Deuti ma enzyme amena magachino mu acid will not work. so enzyme will not work. Also, my germs will not be killed, and also my bones will not be dissolved. So that is. Let us proceed. This is genetics. Sinagunde. Let's skip. This one is coordination. Sinagunde. So tungoita skip. Kamatagai gunda tizi ankato says. This one is also excavator system. Sinagunde. Once you've done that, you'll be able to respond to them quickly. Describe how hard it works. I think for Svetalilongo Sobnobuino, I think it's easier for you to explain how hard it works. If you have a problem, you can come to my inbox. 5 major impact of human activity on environment, climate change. That is so simple to you. Let's see the another is says explain 5 factors affecting primary productivity in fresh water. This is ecosystems Nagundes, but you'll be able to answer them properly. If you have comment, question, come to my inbox. Thank you.